Good afternoon, everyone. My name is Dr. Sophia Wong. I'm an assistant professor of clinical psychiatry at the IU School of Medicine, and I am also the Outreach Recruitment Engagement Coordinator for the Indiana Alzheimer's Disease Research Center. It is with great pleasure I welcome all of you to our first session of this year's Memory University for 2022. And our first speaker this year will be Dr. Jeffrey Dage. So I think the topic he's going to be discussing is going to be a very salient one that has often been raised both by patients and caregivers to me, as well as researchers. And that is, when is there going to be a blood test for Alzheimer's disease? In other words, when do I not have to go through all this extensive neuroimaging, paper pencil testing? Where does the field stand now? And it is with great pleasure that Dr. Dage has just recently joined our Indiana Alzheimer's Disease Research Center last year. And he is a senior research professor of neurology, as well as medical and molecular genetics here at IU School of Medicine. He's now also the co-leader for the GBBS core here at IADRC. He earned his doctorate of philosophy at University of Cincinnati and actually did some really seminal work in blood-based biomarkers at Eli Lilly for over 25 years. And he's going to be talking about that work. And I'm really sorry I won't be able to join you today for moderating the Q&A, but I'm very delighted to actually have another of our outreach core faculty members step up to the helm, and an opportunity for you to meet her as well. She just recently joined last year. Her name is Dr. Nella Pulcinelli. She's a neuropsychologist. She also works at the neurology department at IU School of Medicine. Her primary work focuses on dementia, including Alzheimer's disease. I've really enjoyed working with her as a co-PI on our Leads Diversity supplement, and she has also just been a great pleasure to work with on so many of our OREC projects. So without further ado, let's hear from Dr. Jeffrey Dage.
Hi, and thank you for having me at Memory University. My name is Jeff Dage, and I'm a senior research professor of neurology at Indiana University School of Medicine. I'll be talking about blood tests for brain health and Alzheimer's disease and related dementias. I've been involved in this field for the last 10 years while I was at Eli Lilly and led the discovery and development of some of the tests you'll hear about today. I just joined Indiana University last August, and I'm a co-leader of the Biomarker core within the Indiana Alzheimer's Disease Research Center. First, my disclosures. I'm a minor shareholder of Eli Lilly and Company stock and an inventor on patents associated with the phosphorylated tau assays discussed in this presentation. I've received some support for my research from Eli Lilly, ADX Neurosciences, and Roche Diagnostics. In addition, I have served as a consultant or advisor for Karuna Therapeutics, Genotics, Biotechnologies Incorporated, Gates Ventures, and IslesPath Incorporated. In today's presentation, I'm hoping to provide an overview of Alzheimer's disease biomarkers and specifically address the rapidly advancing blood-based biomarker field in Alzheimer's disease and related dementias. I will cover the tests that are just now being made available as lab-developed tests, as well as those currently being explored in research. Finally, I'm hoping to highlight areas of active research for people to consider leading or participating in the next stages of discovery and development of novel blood-based biomarkers. This is probably not necessary for this audience, but I think it's important to highlight the size and significance of the problem of Alzheimer's disease in the United States. This is information from the Centers of Disease Control and Prevention website. Alzheimer's is one of the top 10 leading causes of death in the United States. An estimated 5.8 million Americans aged 65 and over had Alzheimer's disease in 2020, and it is expected to grow. This disease takes a huge economic toll of hundreds of billions of dollars. But more importantly, it takes a huge toll on the families affected as they try to care for their loved ones. With over 500,000 new diagnoses each year, we need earlier diagnosis and better treatment options. So I'm not sure if you've had this experience in other areas of medicine, but sometimes it can take a long time to get in to see specialists, and then it can take an even longer time to get to a diagnosis. This can happen when experiencing cognitive impairment as well. The path to a diagnosis has many challenges. There are not enough specialists available to provide expert diagnosis. This is a real concern, for when a therapy becomes available, the patients that need treatment won't be able to get an appointment. Even when they get an appointment, the process for cognitive testing and diagnosis can take multiple visits over several years. In this study from Mark Hansen at UCLA, they developed some sophisticated models to evaluate the impact of brief cognitive testing and blood biomarkers on the demand for specialist visits. This would be referrals to neurologists, geriatricians, or neuropsychologists. The graph on the left shows a dashed horizontal line. This is an estimate of the capacity of the healthcare system in terms of dementia specialists. They have a number of assumptions, but demonstrate that without the use of a filter of brief cognitive testing followed by a blood-based biomarker, the system will be overloaded and patients won't be able to get the care they need. You can see in the graph on the right that the model also estimates the wait time for completion of the diagnostic process based on different scenarios. Here you can see again the impact of combining cognitive screening and the blood-based biomarker drops the wait time from 4 to 5 years down to below 1 year to diagnosis. We're not currently fully experiencing the effects these models are illustrating, as disease-modifying therapies are not yet widely available and have had a highly controversial start. But the problem exists, and it will become a crisis for patients once more convincing therapies become available. So why do we need a blood test if we have Alzheimer's disease and related dementia biomarkers now? There are FDA-approved tests to aid in the diagnosis of Alzheimer's disease. Unfortunately, all of these tests have faced some fundamental problems despite being very successful scientifically and in clinical trials. We have had amyloid PET imaging approved by the FDA in 2012, but its use has lagged as it has an expensive procedure and lacks of insurance coverage has limited its use. PET imaging is positron emission tomography and is conducted following the injection of a radioactive drug called a tracer. The tracer, once injected into the patient, will bind to its target, in this case amyloid plaques, and after a brief time, imaging is conducted to measure the amount of signal that is present in the brain. The location and intensity of the signal can be really informative, and the image is usually read by a radiologist with special training. Recently, in 2020, tau PET was approved by the FDA to estimate the density and distribution of aggregated tau neurofibrillary tangles in adult patients with cognitive impairment who are being evaluated for Alzheimer's disease. Unfortunately, it is also expensive and it's limited in geographic distribution, and it's not currently accessible, again, given the lack of reimbursement. Earlier this year, in the month of May, the cerebral spinal fluid A-beta 42/40 ratio was approved by the FDA, also to aid in the diagnosis of Alzheimer's disease. This test has been implemented in individual laboratories and has been used for patient care across Europe and in some larger medical centers. However, with the FDA approval, it is hoped that this diagnostic aid will become even more accessible, but it still remains an invasive procedure, and accessibility as well as insurance reimbursement is still to be determined. So in the midst of all these challenges, we developed assays for measuring phosphorylated tau in blood. There are different sites on the tau protein that can get phosphorylated. In this case, I'm showing you assays specific for either the 217 site or the 181 site. You can see in the graph on the lower right that the two measures are very highly correlated. We demonstrated that these assays can work on more than one assay platform, the Simoa and the MSD, and completed fit-for-purpose assay validations, as well as placed them in a CLIA-certified laboratory at Lilly Research Labs for clinical trial screening. To give you a little flavor for how the assay performed, here's a retrospective diagnostic performance study we completed in collaboration with Adam Boxer at the University of California, San Francisco. This was published in Lancet Neurology just last year. On the left are receiver operating characteristic curves. These show diagnostic performance. You don't have to worry too much about it. The closer the area under the curve, or AUC, is to 1, the better the performance. And you can see that the diagnostic performance to differentiate Alzheimer's disease from frontal temporal lobar degeneration, or FTLD, is high at 0.93 for phosphorylated tau 217. Also, on the right-hand side of the screen, a separation between groups is evident and tracks very well with the presence or absence of amyloid pathology. And you can see that the effect size for the phosphorylated tau 217 is just over 4. We also compared the blood biomarker phosphorylated tau 217 to the gold standard measure of neurofibrillary tangles, tau PET. During screening for a phase 2 study in early symptomatic Alzheimer's disease, we collected 860 plasma samples prior to subjects being referred to tau PET. We measured the phosphorylated tau levels and compared those with the tau PET signal. There was a clear association and evidence of the blood test showing elevations even at low tau PET levels. And importantly, for clinical trial use, there is a very high negative predictive value. This means if a subject was below the cut point on the plasma phosphorylated tau assay, they were almost certainly going to end up negative on tau PET and not be eligible for the trial. So in a very short time, the field has advanced a lot. And in collaboration with Charlotte Tunisen's lab at Amsterdam University Medical Center, we evaluated 6 different assays, all measuring phosphorylated tau in slightly different ways. The key message is that they all worked to different degrees, and the phosphorylated tau 217 assay had the highest fold change, again, just over 4. Many laboratories all over the world are now measuring phosphorylated tau in blood and conducting research studies trying to develop a complete understanding of how it should be used. So I published my first phosphorylated tau 181 paper in 2018, and then in 2020 we made some great advances with the phosphorylated tau 217 assay. These news clips below are just a couple of the mainstream news stories covering this work. Notice the comment on the New York Times piece. Scientists say tests could be available in a few years. And just after 2 more years now, we're getting closer, and there's the adoption of the phosphorylated tau assays around the world. And we're not there yet. There are still other pathologies and other markers, and even other uses for phosphorylated tau that we'll talk about later. So what are the blood biomarkers currently being evaluated in research studies, and how are they associated with things happening in the brain outside of the phosphorylated tau 217 or in addition to? This is a graphic from a recent review from one of my collaborators, again, Charlotte Tunison at Amsterdam University Medical Center. It highlights a few key points that are important to keep in mind when we measure things in blood. What exactly are we measuring? That's important. Where did it come from? And how did it get into the blood? And also, why are the levels different between normal and those with a specific pathology? All 3 of those questions need to be addressed. And on the right-hand side of the graphic, we're going to kind of work backwards, are A-beta peptides, phosphorylated tau, neurofilament light chain, and glial fibrillary acid protein. These are all being shown in blood, and they're the most advanced blood biomarkers. They're also some of the most advanced CSF markers if you move to the left. And we often look for correlations between CSF and blood levels as an indicator as to the specificity for brain disorders, and confirm the idea that what we measure in the blood actually reflects what is happening in the brain. Finally, each of these markers are associated with a specific pathology that occurs in one or more diseases. A-beta peptides are associated with amyloid plaque that primarily occurs in Alzheimer's disease. Phosphorylated tau is associated with tau neurofibrillary tangles, which are also associated with Alzheimer's disease, but can be found in other diseases referred to as tauopathies. Neurofilament light chain is associated with axonal degeneration, and this can occur in many diseases such as cerebrovascular disease, Alzheimer's disease, multiple sclerosis, or tauopathies. Glial fibrillary acid protein, or GFAP, is associated with reactive astrocytes, which can occur in many conditions, including Alzheimer's disease, in response to amyloid plaques. These blood measures can help us develop an understanding of each pathology and how they might interact with one another. As I mentioned previously, plasma measures of A-beta 4240 using mass spectrometry methods have become commercially available as lab-developed tests through a few specific laboratories. Lab-developed tests are not approved by the FDA and are a category of specialty tests run in local laboratories operating under government regulations called Clinical Laboratory Improvement Amendments, or CLIA. I mentioned that earlier. This regulation happens through the Centers for Medicare and Medicaid Services, or CMS. They regulate all laboratory testing performed on humans in the US, except research. And the objective of the CLIA program is to ensure quality laboratory testing. FDA approval usually is focused on the test being safe and effective. The lab-developed tests can be available and commercial without or prior to FDA approval, but they need to be manufactured and conducted in a single laboratory. The FDA has granted breakthrough status for Roche, Fuji-Rabio, and Quanterix for the development of blood-based biomarkers on their diagnostic platforms. This indicates the unmet medical need and the FDA's willingness to help these companies as they attempt to bring their tests to market through the FDA approval process. The blood tests under development include A-beta 4240, phosphorylated tau, and neurofilament light chain. I want to now describe the available lab-developed tests for A-beta 4240. First, the Persivity AD from C2N based in St. Louis. They have implemented an immunoprecipitation mass spectrometry method first developed by the Randy Bateman Laboratory at Washington University in St. Louis. They use a single method to measure both A-beta 42 and 40 to make the ratio. They also measure the ApoE proteoform in the same blood sample, and by combining this with the patient age, they develop an amyloid probability score in patients experiencing cognitive symptoms. The score can fall within one of three ranges: low, intermediate, or high. The figure on the right kind of shows how those distributions will break down across those three ranges. A low APS score is consistent with a negative amyloid PET scan and would suggest something other than Alzheimer's disease being responsible for the cognitive symptoms. An intermediate APS score does not distinguish between the presence or absence of amyloid, and further diagnostic evaluation may be needed to assess the underlying causes. A high APS score is consistent with a positive amyloid PET scan and thus a high likelihood of amyloid plaques. Presence of amyloid plaques in a patient with progressive cognitive symptoms is consistent with an Alzheimer's disease diagnosis. You can see from the graphic on the right how these regions break down in terms of the overlap and proportion of patients in each category. The second lab-developed test is from Quest Diagnostics and is called Quest AD Detect. They also are taking an immunoaffinity purification approach followed by liquid chromatography mass spectrometry. Their method has not yet been published in a peer-reviewed journal. The clinical utility of the method is still unknown, but they claim a cut point of 0.16 and an interpretation such that levels below this are higher than normal risk of having Alzheimer's disease and follow-up testing is warranted. There are likely to be other lab-developed tests to follow for phosphorylated tau and neurofilament light chain. Once these tests gain FDA approval, they might move more into the mainstream and accessibility will likely increase. So to summarize the first part of my talk, we have a huge medical need with over 5 million people in the US suffering from Alzheimer's disease, and as many as 500,000 more being diagnosed each year. There are imaging tools and now cerebral spinal fluid tests to aid in the diagnosis, but they are inaccessible and expensive. Now, the second part of the talk is really for the science-minded viewers, even more so, as we dig into some details about blood-based biomarker research. So let's go over a couple of definitions and background. I mentioned brief cognitive testing, and you might hear about digital cognitive tests as they're mainly research tools and tests using smartphones, tablets, and even some watches, all designed to identify cognitive symptoms as early as possible, such that treatment might be initiated and cognitive function preserved. So right now, they are trying to identify the mild cognitive impairment stage. This is defined through detailed neuropsychological testing. It can be categorized as amnestic or non-amnestic. These are categories related to memory function or other brain functions like decision-making, visual or spatial thinking. For mild cognitive impairment, these have to be observable, but not such that they impair a patient's daily life. Dementia, on the other hand, are the same symptoms, but they have been progressive over time and are at a stage of severity such that the patient's daily life is impacted and deteriorating. Identifying the presence of the disease early can provide us with the best chance of preserving function and delaying or preventing progression. Much of the blood biomarker research is focused on early detection. So those are the symptoms, but what are the causes of these symptoms? The most common is Alzheimer's disease. This was first described in 1906 in a patient that died after experiencing progressively worsening dementia symptoms, just like I described. Dr. Alzheimer identified amyloid plaques and neurofibrillary tangles in her brain. Since this time, thousands of brains have been evaluated in both early onset, where symptoms start before 65, and late onset, which starts after age 65. And we know now that the plaques include peptides from the amyloid precursor protein called A beta 42, and the neurofibrillary tangles have hyperphosphorylated tau protein. The field has also identified some rare genetic causes of early onset Alzheimer's disease. These are attributed to mutations in the amyloid precursor protein, or APP, or the presenilin gene, which is a protease responsible for processing the APP protein. Interestingly, in Down syndrome, there's an extra copy of chromosome 21, which happens to contain the APP gene. This additional copy results in the early development of Alzheimer's pathology. These rare genetic causes provide clear evidence that amyloid proteins are a cause of this progressive disorder and can be very useful in learning about the utility of blood-based biomarkers in Alzheimer's disease. So why is it Alzheimer's disease and related dementias? We have that added on. So what are the related dementias? Well, early cognitive changes I mentioned a few minutes ago that often lead to dementia can be associated with a variety of different and often overlapping pathologies or causes. And when a patient experiences early cognitive changes, doctors often want to look into the possible causes that may or may not be reversible and might require referral to specialists other than those specializing in dementia diagnosis and care. And these can come from family history, blood tests, and imaging, either with magnetic resonance imaging, known as MRI, or a computerized tomography, known as CT imaging. There are many conditions that can lead to cognitive changes, and some, like I said, are reversible. However, there are also pathological proteins that can be identified in the brain that are thought to be responsible for symptoms. I mentioned amyloid plaques and neurofibrillary tangles or threads made of tau protein, but in the majority of cases, these two pathological proteins are not alone. Often, upon postmortem analysis, there are Lewy bodies made of alpha-synuclein or TDP43 inclusions, or even quite often signs of cerebrovascular disease as well as specific brain regions of experiencing atrophy. So all of these can be indicators of a complex sequence of events that may have led to dementia and untimely death. So what is a biomarker? The title talked about blood tests. These are occasionally used interchangeably, but biomarkers are usually used to research, while tests are used in clinical practice or patient care. I'm talking about both. I'm going to show mostly research biomarker data, and I only highlighted just a couple of new blood tests. Since the data I'll share is mostly that research, I'm going to stick to the terminology of biomarker as much as possible. So a biomarker is an objective measure that captures what is happening in a cell or organism at any given moment. They can be used to understand fundamental biological processes, study pharmacodynamics, which is the action of a drug, and can even be developed together with a therapy or as a standalone test. Biomarkers can also serve as early warning systems for your health or inform on what has happened. Consider simple biomarkers we each experience often, so like body weight or blood pressure, and how we track these measures as a sign of our health. There are blood tests for cholesterol also that inform on heart disease risk, the leading cause of death. Because we know identifying the risks early make it easier to address and prevent disease. And today I'm talking about A beta 4240 or phosphorylated tau for Alzheimer's disease in that same kind of context. Can we use them to identify disease early so that we can start treatment or change our lifestyle? So there are actually many biomarkers for Alzheimer's disease, and they're all at various stages of development. I mentioned some of these already. One is MRI, but advanced imaging techniques can accurately pinpoint signs of atrophy in the brain. There are also several kinds of PET imaging or positron emission tomography that can be useful in Alzheimer's research. I didn't mention FDG PET, but this is fluorodeoxyglucose, and it can be used to look at metabolic activity or the loss of that activity. I mentioned amyloid PET and tau PET, which allow for the direct measurement of these pathologies in the brain and allows us to measure both the amount and the location. Both of these have now been approved by the FDA as tests to aid in the diagnosis of Alzheimer's disease. And there are also genetic tests for the rare forms I mentioned earlier, but also for the risk variants like the ApoE gene. Then there are the biofluid biomarkers, either in cerebral spinal fluid or blood. As I mentioned just this month, the cerebral spinal fluid test for A beta 4240, developed by Fuji Rabio, was approved by the FDA to aid in the diagnosis of Alzheimer's disease. And of course, I'm going to be talking the rest of the time, hopefully, about the blood-based measures. So sometimes it's helpful to take a long look back in time and see how things developed and have changed. All of these biomarkers have made large contributions to our understanding of Alzheimer's disease, as well as the clinical trial design for investigational therapies. Many of these biomarkers are used first in clinical research for many years before advancing as diagnostic tests. And you can see that with this development on the left-hand side of the screen of amyloid PET in the early 2000s. And that was with the Pittsburgh compound B or PIB. Yet the FDA approval did not come for any amyloid PET tracer until 2012. Tau PET was initially published, I think, in 2009, with the FDA approval not coming in until 2020. In each case, it took a decade to bring these tests to patients. A lot of work goes into characterizing these biomarkers and understanding their clinical use. CSF amyloid biomarkers have been around longer, but being able to connect the CSF A beta 4240 ratio with amyloid PET imaging in 2012 is what really led to the recent FDA approval. And it's often the case that a breakthrough in one area leads to additional advancements in others. And we're now using both PET and CSF testing in our evaluations of blood biomarkers and their potential clinical utility. For this approach to be successful, it's critical that we understand how they relate to one another. And that is a big driver behind the ATN framework that's in the upper right. And that was included in the Alzheimer's Association National Institute of Aging 2018 research criteria for Alzheimer's disease. They focused on PET and CSF testing mostly in that report, but it was published the same year that we published our original paper showing the utility of measuring phosphorylated tau in blood, which is in the upper right corner, in terms of its relationship to both amyloid PET as well as cognitive function. Now, the ATN framework started with this back probably earlier, but with this 2012 hypothetical model, which was presented by Cliff Jack from the Mayo Clinic. He proposed an order of biomarker abnormality that precedes clinical symptoms. The order was one in which amyloid pathology was first, then followed by tau, neurodegeneration, and then came changes to cognition. This hypothesis has been at the center of Alzheimer's research for the last decade and has undergone many refinements in terms of the numbers of years and the relationship of each pathology in terms of time. The key feature of pathological evidence being detected many years prior to cognitive symptoms has stood up to many experiments in late onset, early onset and rare genetic forms of Alzheimer's disease. Some think this reflects poor tools to track cognitive changes. So there are many research projects trying to develop better tools. Others believe there's an element of brain resilience to the presence of pathology and are exploring what might be responsible for imparting such resilience. The key for me is that all of these areas of research would not be possible without first having reliable biomarkers that can report on the presence of the pathologies of Alzheimer's disease, amyloid plaques, and tau neurofibrillary tangles, and an understanding for how these relate to one another. So from the hypothetical model by Cliff Jack in 2012, here I'm showing data from the first published biomarker study where we measured phosphorylated tau 217 in blood. And this was published in 2019, where I collaborated with a group at Lund University in Sweden. This was Oscar Hansen. And they have a long-standing biomarker study called the Swedish Biofinder Study. It stands for Biomarkers for Identifying Neurodegenerative Disorders Early and Reliably. It's a large longitudinal study following 1,600 subjects, and they collected cerebral spinal fluid, plasma, and conducted PET imaging. This is very similar to our own Indiana Alzheimer's Disease Research Center, as we get more people to participate in the cerebral spinal fluid, blood collections, and imaging. In these figures, we measured biomarkers in blood and cerebral spinal fluid and developed data-driven models to try and understand the relationship of the biofluid biomarkers to one another and to amyloid PET imaging, which is plotted on the horizontal x-axis. The PET imaging increases over time, and the dark red dotted line would mark the threshold at which a subject would convert to positive. And you can see in the lighter colored dotted lines the points at which the biofluid biomarkers first change their trajectory. The ordering is very similar to the hypothetical model proposed by Cliff Jack, with some interesting differences. Both the cerebral spinal fluid on the left and the blood measures on the right are changing ahead of PET positivity threshold. But they continue to change long after. This suggests that the temporal ordering of the pathologies might be a function of the biomarkers that are used, since PET is measuring the insoluble pathology in the brain, while the biofluid biomarkers are measuring soluble forms that are associated with the pathologies. The changes in the biofluids might be easier to detect and thus show earlier signs of disease. Another interesting difference is between the CSF and plasma biomarkers. The CSF measures seem to plateau, while the measures in blood continue in an upwards trajectory. This might indicate that the blood measures could be more informative when measured at annual visits to track change over time. In another study with two blood-based biomarkers, phosphorylated tau 217 and neurofilament light chain, or NFL, we sought to understand how early these markers might change prior to symptom onset. As I mentioned earlier, subjects with rare genetic mutations that cause early onset Alzheimer's disease can be very useful to understand the relationship between biomarkers, as well as understand how early changes might be indicative of future symptom onset. That is exactly what we did here in collaboration with Eric Ryman at Banner Health in Arizona. This study is in 622 subjects, with 59% of them carrying mutations in the presenilin 1 or 2 genes, as well as 41% non-carriers for comparison. The Colombian Autosomal Dominant AD Registry has demonstrated a median age of symptom onset of 44 years, and it's indicated by the vertical dashed line. I hope it's clear that both the phosphorylated tau and neurofilament light chain change prior to symptom onset compared with the non-carriers, and that the phosphorylated tau increases only in those subjects that carry the mutations. This would suggest that we can detect abnormal changes in phosphorylated tau protein in the blood many years prior to symptom onset. Now, in another collaboration with the Longitudinal Investigation for Enhancing Down Syndrome Research Study, or LIFE-DSR, we confirmed our findings from presenilin mutation carriers that the biomarkers change prior to symptom onset, only this time we looked at subjects that carried an extra copy of the APP gene. In Down syndrome patients, we measured two kinds of phosphorylated tau, the 217 and 181, as well as neurofilament light chain. We also measured additional blood biomarkers, the A-beta 4240 and glial fibrillary acid protein, or GFAP. Again, we see changes occurring prior to anticipated symptom onset, where some of the biomarkers are flat or plateaued and then become abnormal. In the case of the A-beta 4240 ratio, it decreases with pathology, and phosphorylated tau and GFAP increase. Neurofilament light chain, once again, is consistently changing, but without a control group, the importance of this change is not able to be determined. This age-dependent change in neurofilament is a complicating factor with this measure and its use in many diseases. So what about in the general population? So in a collaboration with Michelle Melky in the Mayo Clinic Study of Aging, we measured plasma phosphorylated tau in 1,329 participants. The goals of the Mayo Clinic Study of Aging are to develop tools to predict and prevent cognitive decline in dementia, develop risk prediction models for cognitive impairment, and conduct aging-related research to promote successful aging. Now, this is a population-based study. That means participants are selected at random from the population of Olmsted County, Minnesota, and asked if they'd like to participate. The majority of participants are cognitively unimpaired, with only 153, or a prevalence of 13%, MCI. So aged 60 and over. So you can see that starting about age 60, an increase in the proportion of amyloid positive subjects by PET imaging. And this corresponds to increased levels of phosphorylated tau in the blood, shown in the red. The amyloid positives in the red and the amyloid negatives in the black. And this is similar to what we've observed in the Colombian autosomal dominant AD registry and the Life DSR studies. And it supports the idea that the blood biomarker is elevated prior to symptom onset, even in the population-based study. Now, with a study this large, it also enables us to evaluate medical records for the diseases that these participants might be experiencing. And this is important in population studies, and it's an important step in biomarker research, as we need to see what factors might influence the levels and how these should be considered when patient results are available for interpretation. There are a number of items that don't overlap with zero, indicating a significant association. You can see education, diabetes, dyslipidemia, and hypertension and cancer are all associated. But once you correct for age and sex, these are no longer significant. This kind of indicates that the imbalance in age and sex are likely explain these effects. Likewise, the effect of the risk variant, ApoE, is significant until results are adjusted for amyloid PET. And this fits with the theory that ApoE, or the E4 risk variant, is associated with amyloid pathology. Now, even after adjusting for age, sex, and amyloid PET, several diseases remain significant, including body mass index, smoking, chronic kidney disease, stroke, and myocardial infarction. Some of these are easy to understand. For example, as body mass increases, so does the blood volume, and the brain stays the same. This results in lower levels of phosphorylated tau in the blood coming from the brain. In a similar manner, subjects with chronic kidney disease have impaired clearance of many proteins that normally would rely on the kidneys for clearance. And this reduced clearance results in increased levels of phosphorylated tau. But stroke and myocardial infarction are more difficult to explain and may be contributing factors to the cognitive symptoms on their own, or potentially acting in combination with Alzheimer's disease pathology. So studies more closely investigating the relationship between Alzheimer's disease pathology, stroke, myocardial infarction are going to be really important as we develop new markers for specific for cerebrovascular disease, and then looking at how these relate to one another. So the initial cross-sectional studies in presenilin, Down syndrome demonstrate some key learnings, and they create hypotheses for how we might be able to use blood biomarkers. And we're able to show similar results in a community study and identify some of the comorbidities that need to be considered when interpreting biomarker results. This is the first area of really active research I wanted to highlight. And as now we need longitudinal studies where we have both control groups and those increased risk are needed for participation. And these can be focused on the autosomal dominant carriers and non-carriers, but can also more broadly include just risk like family history or ApoE carrier status. And there are current ongoing studies like the longitudinal collections within our own Indiana Alzheimer's Disease Research Center, and these can be extended into additional community-based cohorts. Lastly, to summarize the science and highlight some additional opportunities for future research, I really like this graphic. It reminds me that the pathologies are actually very heterogeneous. We talk of A being for amyloid, but actually amyloid pathology can exist in the brain as diffuse, compact, or dense cord plaques. Additionally, it can show up in blood vessels as cerebral amyloid angiopathy. When we measure A-beta 4240 in the blood, we associate it with the PET signal, and PET does not easily differentiate or identify pathology subtypes, and there is significant diversity among patients. Similarly, for the T or neurofibrillary tau tangles, there can be threads, tangles, as well as tangles associated with neuritic plaques. There's a lot of heterogeneity in the pathology and also in the binding characteristics of the different tau PET tracers. These differences will need to be explored in large comparison studies to understand how to best use these tools to help patients. Now, the N is for neurodegeneration, and it's the easiest to see with extensive brain atrophy over the course of Alzheimer's disease. But this occurs over 10 or more years, and the amount of change in the blood at any one time might only be an indicator of the rate of change. But again, much more research is needed, especially in understanding the influence of comorbid conditions on the rate of neurodegeneration. Now, we need to do more studies to understand all of these relationships in greater detail in patients where we have collected blood samples, performed PET imaging, and where families have elected to participate in brain donation programs, like what we have within the Indiana Alzheimer's Disease Research Center. Now, according to this review article from Charlotte Tunison at Amsterdam University Medical Center, there are 5 phases in the development of biomarkers to aid in the diagnosis of Alzheimer's disease. Starting with preclinical exploratory studies and moving through the implementation and impact on clinical outcomes. You can see from the color coding, we're moving into phase 4, which is prospective studies and real-world performance. There's never been a better time to get involved in Alzheimer's disease research and to help advance the development of blood tests for brain health in Alzheimer's disease and related dementia. And finally, my contact information, and I'm available for questions and discussion. And thank you very much for your attention.
All right. Hi, everyone. Thank you, Dr. Day, for giving that fantastic talk. We are extremely fortunate to have you here for the first session of our Memory University. I know some folks have already put questions in the chat. I know we are running low on time, unfortunately. Again, apologies for the technical difficulties we had earlier, but we will try to get to a couple of questions, and then any follow-up questions that you guys have, we'll give you our email address to reach out to us. So one of the questions that came up from one of our participants today is, and a couple of the questions are related, but if a person has dementia, how often should they be getting these blood tests, repeatedly, once, that kind of thing?
Yeah, that's a great question, the timing. So most of the research has been focused on early detection, and there hasn't been a lot of research on once a person's got the diagnosis of dementia, what is the utility of the measurement. And so right now, it's probably best once you're diagnosed that you don't need to get retested. And I think it's not the kind of thing that we have enough research done to tell you that it'll add more value. I think when it comes to early detection and you're negative, there we see pretty slow increases over time. And so, annual visits every couple of years, things like that are what people talk about. And it's just about keeping track of risk. or seeing if Alzheimer's pathology is developing. But most of the time, people are even all of the available tests are used in conjunction with symptoms. And so when a person's being evaluated for the possibility of having dementia, so it's in the context of symptoms. And all those early things are still all in research as it pertains to risk.
So that kind of preempted my next question, which is, if you're negative, does that change the story a little bit? Which it sounds like it does.
Yeah, if you're negative, you'll want to. You can follow up, every year, every couple of years, and make assess your level and how it's changing over time.
Mm-hmm, mm-hmm. Another question, sort of related to that, is if it's positive. And maybe there are no symptoms, so maybe not dementia, but you do have a positive blood test. What does that mean, or where do we go from there?
Yeah, we're just now getting more into this, the research associated with risk, and what degree of risk does it confer to be elevated, because we are showing that we can detect it ahead of symptoms. But again, like right now, all of the labeling and everything for all of the FDA approved biomarkers are all in the context of a person presenting with symptoms. And so it's all research related to risk when you don't have symptoms. And that research is ongoing, and that level of risk that these elevations confer is still being investigated.
Mm-hmm.
And so it is, I think it's a really important area of research to get involved with, and to see how we can use it to improve patient care and early detection and treatment once those become available.
Absolutely. I mean, I think it's a very exciting technology to have, or a very exciting test to have, especially as you described it, in terms of there are a lot of very real limitations on some of the tests that we use right now to help these diagnoses. So we are going to, I know that there have been additional questions, and I apologize to everyone. You guys have had great questions. Please, we're going to stick around for another minute or two. We do have some just announcements to make, and also you'll have our contact information. Please follow up with us through our email, as well as on Facebook, and send us your questions, and we'll answer them to the best of our ability, or connect you with the people that can answer them. So thank you, everyone, and just stay tuned for one more minute for the announcements.
Yeah, thank you very much.
I want to thank Dr. Dage and Dr. Postinelli for a really wonderful session to kick off this year's Memory University. You'll be receiving a survey after the webinar, so please take a few minutes to share your feedback. This will be really important as we try to develop future topics for Memory Universities in the upcoming years. And thank you so much for coming. We also want to remind everybody that you can stay in touch with our center through email if you have any questions. And you can also call us at 317-963-5500. Check out our website, and we're also on Facebook and Twitter. And you can contact us if you have questions about how to obtain a clinical referral for a loved one, or if you have questions about how to participate in many of our research studies at our center. I also just want to remind everybody that this session is being recorded and will be available on our Facebook page if you wanted to see it again, or share it with a friend who didn't have the opportunity to join us. And we have 3 other upcoming talks I hope you can join us for. The next talk on Friday, June 10th, will be with Anita Harden, Common Misconceptions About Power of Attorneys. And then on June 17th, for the live session, there will be CME available for an exciting talk with Dr. Robert Russell, a geriatrician, also sits on the advisory board for the Alzheimer's Association, understanding the challenges of delivering Alzheimer's care in minority populations. We're very grateful to the Indiana Geriatric Society for sponsoring this talk. And then finally, I'll be giving an update on what is the latest information on Friday, June 24th on long haulers. What do we know now about cognitive symptoms after COVID-19 infection? Again, we want to thank our sponsor, Sokoa, for this. So once again, thank you so much for joining us for our first session of Memory U. We look forward to seeing you for the upcoming 3 sessions, and have a wonderful afternoon.